All right, well, let's go ahead and get started. Welcome everyone to this evening's lecture. The Science Museum lecture series is brought to us by the Sheridan College Museum of Discovery, which has had the lecture series going for over 20 years now. My name's Scott Newbold. I'm a member of the biology department at Sheridan College and help coordinate the lecture series. Before we introduce tonight's speaker, I just have one short announcement, and that is that in mid-April, I don't remember the exact date, I apologize, we have the next speaker who is a faculty member at the college in the biology department, Laura Hassman, talking about bioassessments and toxicology of stream systems. So again, that date will be coming out soon. All right, tonight for our lecture, Georgia Knaus has graciously agreed to join us and provide the introduction for our speaker. She's the lead paleontology program manager at SWCA here in Sheridan, SWCA Environmental Consultants. So thanks, Georgia, for being here. Appreciate it.
Yeah, thanks, Scott. Good evening, everyone. Sorry about that feedback. So yeah, we're really excited to have Daigo with us this evening, and I guess we'll get a little bit of his paleontological background. He's had a variety of it since he started. And the reason I'm here is because he's actually worked a bit as a mitigation paleontologist for our company, as well as for a couple others throughout Wyoming, Utah, little maybe in Colorado, Montana. But he came to the United States in 2004, actually from Yokohama, Japan, to do his undergraduate work at the University of Montana in Bozeman. Montana State University, in Bozeman. And he liked it so much, he stayed there for his master's degree. And currently, Daigo's working as a science faculty at the Miles City Community College. But it's very clear if you know Daigo, he's enjoyed teaching since he's, I think, started in paleontology. So even as a master's student, he was already starting to mentor undergraduates in science and education and learning all that they could about different types of paleontology. Then he went on from Montana out to Arkansas to university to get his Ph.D. And I think it's interesting people always ask, what do paleontologists do? And I think tonight you'll learn a little bit about what a specific kind of paleontologist does. Daigo has experience with vertebrate fossils, and that might be what you think of when you think of paleontology a lot, dinosaurs, T-Rex. But he also knows a lot about chemistry, and that aids in his, especially when he works with us and when he does field work, he really understands the taphonomy, how the fossils get there, depositional environments, and kind of understanding why and where you may find different kinds of fossils. And so we're really excited to have him talk to us tonight about his work in Utah. When he's not doing paleontology, Daigo likes to run and backpack, which are really important for some of the field work he's done in Utah and the Kaiparowits, which is very, very rugged terrain. And hopefully we'll get to see a few photos of that this evening. And he also likes to play the guitar, which I'm guessing he probably did a little bit of when he was in the field. So his talk tonight is on the Kaiparowits Formation, and I think I'll turn over to Daigo.
Great. Well, thank you so much for the opportunity again. And I'll just share the screen right now. Okay. Right, so yeah, this first slide actually shows some topography in the Kaiparowits Formation right there in Grand Staircase Escalante National Monument. But really, I kind of geared my talk today for more on like a general education chemistry side of the story, right? So I will talk a little bit about introductory chemistry stuff and how it relates to this large, broader picture of paleontology, right? So yeah, having said that, I'm gonna move on to the next slide. And I always kind of run out of time, so I just put the acknowledgement in the second slide here. So obviously, Georgia and Scott for the opportunity. Thanks again. And also, majority of the work is kind of continuation of my PhD work. So I would thank my PhD advisor, Selena Suarez, and also the contributor from the Bureau of Land Management down in Kanab, Utah, right? Alan Titus is the paleontologist in the Bureau of Land Management in Utah, and some dinosaurs are actually named after him. So he's pretty big deal down there. Also, Eric Pollock and Lindsay Conway for the lab assistants, and Hunter and Thea helped me with the sample preparation. So they are the undergraduate students I mentored while I was in a PhD program. Right. So having said that, let's move on to the talk tonight. Right. So Cretaceous. Some of you might be familiar with what the Earth was like during that time. It's really exciting time in terms of paleontology, evolution, and paleoclimate. Right. So the first thing first. So the land plants had breakthrough in terms of their evolution. So the first flowering plants evolved during the Cretaceous, right? And following that evolution, we started seeing all the pollinating beetles, right? So, that happened during the Cretaceous time, right? And also on the other side of the biology, the vertebrates, right? We have not marsupial, the placental mammals actually evolved during the Cretaceous as well. And following that, we also had the evolution of modern birds, right? So really, Cretaceous is this transition from the typical Mesozoic dinosaur kind of dominated ecosystem toward more like the ecosystem that we know today, right? So again, for that, I would say Cretaceous is, particularly like late Cretaceous, is one of the most exciting time in terms of the evolution and ecology, right? And obviously, the very last moment of the Cretaceous, we had some asteroid hitting Earth to wipe out the dinosaur, right? I'll come back to that point if I have enough time, but let's move on, right? And also in terms of geology, the North America looked a little bit different, right? So the Rocky Mountain, the rise of the Rocky Mountain started around the early Cretaceous, right? And that thin crustal deformation called severe orogeny kind of changed to more like a deeper crustal deformation called laramide orogeny around late Cretaceous as well, right? And also on this picture, you can see this Western Interior Seaway covering majority of the North American continent, right? So the landmass was actually subdivided into this portion called Laramidia and Appalachia, right? So, but this Western Interior Seaway retreated by the end of the Cretaceous about this much, right? So we only had small amount of the sea left at the end of Cretaceous, right? Right, so that's enough background for the paleontology and geology, I say. And yeah, we'll kind of focus more on the actual study subject now, right? So the Campanian is second to the last piece of Cretaceous, right? So toward the end of the Cretaceous, but what's unique about this time period is the elevated biodiversity, right? So these figures, two figures from two different papers, but they're showing this really highly diverse groups of horned dinosaurs, the ceratopsids, right? So this one is the chasmosaurine, so the one with the large frill, right? We had 7 or so different species across the United States, right? And in terms of the short frill version of those horned dinosaurs, we had a lot more diversity, right? With all the different frill shape and all the different horn shape, right? And among those, various dinosaurs, we actually had this 2 chasmosaurine, the Cosmoceratops and Utaceratops, as well as 1 centrosaurine, the Nasutoceratops, right? So all these 3 different horned dinosaurs existed in this particular formation called Kaiparowits Formation, right? So that's the rock formation I studied for my PhD and following study, right? So really diverse ecosystem, right? And also another reason why we chose this particular formation is the previous study about stable isotope geochemistry, right? So there's this whole study done globally, and they compared all the isotopic composition of some different fossil material from this amyl study 2004 and 2006, right? So if I get some data, I have something to compare to the global scale study. So that was particularly kind of fascinating to me. So that's the reason why I chose this time period, right? And really, the research question, I can phrase it in many different ways, but is to really contribute to this understanding of this unique time slice of the Earth's history, right? So what supported such biodiversity during the Campanian is the big picture question, right? But to study that, we can investigate the fossil record as well as paleoclimate and paleogeography, right? It's really impossible to do everything on my own. So what I chose was the stable isotope geochemistry, the big word on the title, right? So I'm going to explain a little bit about how this works, and I will talk about the actual application in the paleontology, right? Right. So you may remember this from either like a college chemistry class or high school chemistry class, but the isotope, the word isotope means you have same elements, meaning the number of proton is the same, right? So again, the number of proton really decides what kind of chemical property the atom can have, right? But the difference between different isotope is the number of the neutron, right? So you can say that isotope is something, same element with the different number of neutrons, right? So in terms of the nitrogen, we have nitrogen 14 and 15, right? So what that means is we have 7 proton and 7 neutrons on the nitrogen 14, right? But nitrogen 15 would have 1 extra neutron, so 7 protons and 7 neutrons, right? So that's the isotope, definition of the isotope. And as you probably know, some isotopes are stable and some isotopes are radioactive, right? So the stable isotope is the kind that does not decay through various processes. But this radioactive isotope, on the other hand, in case for the carbon, 12 and 13 are stable, but this carbon 14 is radioactive. So one of the neutrons in a nucleus would emit the beta particle or the high energy electron, right? And this neutron would turn into a proton, right? So, 1 negative charge is emitted, so that turns into positive charge, right? So yeah, that decay process would create nitrogen 14 as a result, right? So this process takes about, half the carbon 14 would decay in 5,730 years or so, right? So that's the basis for carbon dating, right? Which is not particularly useful for my study because the rocks and fossils are much older, but those are very important too for archaeological specimens, right? Right. And in terms of those isotopes, they naturally exist in the natural world, right? So, and we have these known ratio between these different isotopes already studied, right? So in terms of the chlorine, about 76% of the chlorine has the atomic mass of 35, right? And about 24% of the chlorine would have the atomic mass of 37, right? So multiply by those ratio, added them together, you can get this 35.5 as the average mass of the chlorine atom, right? So these are the numbers you see on the periodic table, right? And often in this stable isotope geochemistry study, we communicate these numbers with the delta notation, right? This is a little bit complicated math, but the idea is the ratio of the sample is compared to the known standards, right? And we use this per mil notation, which is same thing as part per 1,000, right? So the resulting notation for the stable isotope would be, delta 18O equal -10 per mil, something along that line. And what this notation means is this sample has the ratio of the stable oxygen 18 10 per mil lower, so in other words, 1% lower than the standard mean ocean water, right? So that's what the notation really means. But for the sake of this talk, you don't really have to understand the entire definition of that. You should be able you should be able to tell by comparing those numbers, which number is higher and which number is lower. And that has something to do with this stable isotope ratio, right? So that's all you need to know for this talk. Right, and another thing to note here, so this stable isotope ratio can actually change due to the physical and biological processes. So by analyzing this ratio, we can kind of interpret what kind of process took place or what kind of metabolism they had in those animals, right? So for example, evaporation preferentially removed the oxygen 16 over oxygen 18. And to understand this phenomenon, you can kind of think of those two different isotopes as like lighter isotope and heavier isotope. And if something is heavier, it's easier to settle down and it's harder to remove, right? So it exactly works that way in this isotope, right? Oxygen 16 is lighter, so they preferentially evaporate, and the leftover water would have more oxygen 18, right? So that evaporation would increase the oxygen 18 ratio in the leftover water, right? And another practical use is the carbon 13. So, again, the heavier carbon 13, would settle more readily than the carbon 12, right? So in terms of our metabolism, we will keep accumulating, well, not necessarily keep accumulating, but the biological tissues tend to have heavier or higher carbon 13 concentration compared to the food they are eating, right? Right, so again, the practical use of carbon 13 is then tracing this or investigating this trophic relationship, right? So the carbon 13 accumulates in the biological tissue. So by using this relationship, right, we can generalize this relationship between the producer, primary consumer, and the secondary consumers, right? Right, so that's the very useful tool for studying the paleoecology, right? Then the oxygen, on the other hand, is often used for the hydrology, right? And also by comparing different metabolism of different animals, we can extend this data to investigation of the temperature, right? So that's the main use of the O18 in paleontology. So precipitation and drinking water, habitat water, those can be studied by looking into these isotopic composition. And one important thing to note here is the bone mineral, right? So the bone is the actual material I'm studying. And they have, bone crystals have carbonate and phosphate in their system, right? So now I'm gonna have to talk about what they actually mean here, right? Right, so again, the bone mineral, so those are tiny crystallites of bioapatite embedded in the collagen matrix, right? And often people use the word like carbonated hydroxyapatite or bioapatite, anything along that line. And what that really means is our bone mineral is primarily apatite, which is calcium phosphate, but some of those phosphate ions are replaced by carbonate ion, right? So in terms of weight percentage, we have about 8% carbonate in our bone apatite, right? So in a sense, this is not pure apatite, right? That's the reason why we use the word carbonated or bio or anything along that line to distinguish these from those pure mineral, right? And along that line, I need to also mention this bone apatite is very chemically diverse group of mineral, right? So this calcium ion can be replaced by some cation like a barium or sodium, potassium, any of those, right? And there's always the tiny amount of those replacement happening. So in a sense, bone is kind of important storage for our chemicals and nutrients, right? As you probably know, birds, right? The female birds dissolve some of those femur bone and use that calcium for precipitating eggshell, right? So yeah, bones are really dynamic system in terms of the chemistry. And that kind of makes things a little bit harder for us to interpret in terms of the fossil study, right? Right, and also another associated issue is the alteration. So because they are so chemically versatile, that makes them really unstable as well. So before we use these chemical signals from the fossils, we have to make sure those signals are actually preserved biological signal, or it's just a signal from the alteration process, right? So the diagenesis, the word diagenesis means the sediments turning into rocks as well as bones turning into fossil, right? Right. So to rule out that possibility of the contamination, we can use the petrography. So this is the microscopic slides of the bone. So the first picture right here shows this kind of odd lines here. And by using the cross-polar light, you can actually see this different mineral phase, right? So if you see something like that, there's a high chance of like at least these portions recrystallized minerals, right? So if we get the chemical signature from these particular locations, we would probably have non-biogenic diagenetic signal instead, right? So we need to avoid this kind of situation. If the bone's pristine, nicely preserved bone, then what you usually see is the microstructures still preserved in the bone, right? So that's something you can see again with the cross-polar light. So this nice layering shown in this cross-polar light is one way to say the structures are well preserved. So chemical signature is somewhat reliable compared to the situation in the previous slide, right? And along with all the dinosaur bones and teeth, another thing I used is the turtle shell. And the turtle shells also have this kind of nice crisscross type extinction in the polar light, right? So again, by finding this kind of fabric in a fossil, I can actually say this is probably okay to use, right? But after getting all the information, I have to compare these signature, with each other, as well as potential, diagenetic condition. So if something really stands out as really high isotopic composition or, contamination with some other ions, such as silica saturation and stuff like that, we can actually rule out those to be biogenic signal as well. And lastly, the more mineralized the tissue is, the higher chance of biogenic signal preservation. So often in this kind of study, tooth enamel or the Gauss scale, those are highly mineralized tissues, so they tend to preserve the biological signal more readily than spongy bones, right? Right. And also, after getting all those biogenic signal, then what you have to do is back calculating the composition of isotopic composition in the water they are actually consuming. So all the equations, you don't have to know the detail, but what I'm showing here is actually derived from the study of modern animal, right? So by observing isotopic composition of those modern bones, they came up with these conversion factors for fossilized animals, right? There's some degree of uncertainty in terms of dinosaurs because we don't actually have the dinosaur, but these are based on some closely related organisms, right? Namely birds, right? Right, so these are my tools to, again, study this paleoclimate of Kaiparowits Formation, right? Right, so let's finally, it took a while, but let's talk about my case study here, right? So the tyrannosaur teeth associated, and that's associated material from the rainbow and unicorn quarry of the Kaiparowits Formation, right? So this is the paper I published not too long ago. And it is kind of interesting thing to see my own publication because, that was actually published on the journal in England, right? So all the English, or not English, like the words like paleontology or fossilization, those are all edited to British English, right? So the paleontology has one extra A, and the fossilization has S instead of Z and stuff like that. So it was kind of nice to just like read it and notice all the differences. But jokes aside, right? So the main idea was comparing these isotope data to previously studied information, right? So the mean temperature was estimated by using plant fossils, right? So all the margin of the leaf, how jagged it is, actually tells you about the climatic condition, such as, that's the humidity portion. And the stomata's concentration will also tell you about the atmospheric condition. But this Miller and others from Denver Museum did this previous study and estimated the temperature as around 20.2 degrees Celsius. And what you need to consider here is the latitude of southern Utah during the Campanian was a lot higher. Again, the continents are moving because of the plate tectonics. So back in those days, our continent didn't sit in the exact same location, right? So the latitude was higher, but the temperature was this high. Then what you need to think about is the greenhouse condition during the Cretaceous. The CO2 concentration was a lot higher. Some studies even estimate that to be 1,000 ppm or so, which means it's about 2.5 times higher than today's CO2 concentration, right? So the global climate was quite different, right? But what I'm more interested in is the particular local climatic conditions. So I wanted to see some seasonal variation in their ecology, right? So then I decided to use the tyrannosaur tooth, right? Because this tyrannosaur teeth, they often shed the teeth, right? So this climate record recorded in single tooth is somewhat of a shorter duration instead of a like lifetime, which is in our situation, right? We shed tooth only once, right? So the chemical signature in my tooth now is about like averaged information about last 30 some years, right? So that's not going to be very useful, but the dinosaur teeth actually has a lot higher potential than that. Right, so having said that, these are the actual specimen I used in my study, right? So the shed teeth, I acquired 3 of them. These 3 teeth are from the tyrannosaur, like carnivorous dinosaur, right? And associated material from the same quarry was also analyzed to kind of make broader context in terms of the paleoclimate, right? And again, the technique I use is the stable isotope geochemistry. So I analyzed the carbon and oxygen isotope composition in the bone carbonate, as well as the bone phosphate, right? I'll talk about the reasoning a little bit more in detail later. Right, but this is the map of the study site, right? So this is the United States right here, and Utah's right here in the south central portion is expanded in this picture right here. So right around here is the quarry. So in order to get there, you have to take the Highway 89 and just go into this dirt road and drive quite a bit into it, right? So this East Kaiba monocline is the major geologic feature, and all the rocks are almost vertical. So this middle picture right here is the good representation of this East Kaiba monocline, right? And once you cross this jagged ridge, then you can get into this nice badland topography, quite high, really rugged geography. But yeah, so that's the main study site. And you can see most of the sedimentary rock layers are near horizontal, right? So the tilt is not that extreme for the rest of the formation, right? Just something along this monocline is pretty high, but everything else is pretty flat. And this last picture on this slide shows some fossils during the excavation, right? So this is where exact location where my samples came from. Right, and to make like a better connection with what kind of dinosaurs you can find, I just put this picture right here. So this is the Cosmoceratops on the number, the letter A right here. And this is the Utaceratops, B, and the C is the Pseudoceratops. They recently reported this new dinosaur with the armored scutes. So those armored dinosaurs was also found in the same formation. And most notably, this relative of T-Rex was found in there as well. Right. Right. So this would be the graphic represent or artistic rendition of the Kaiparowits Formation. So pretty green, right? And they have multiple dinosaur species, including this horned dinosaur, 2 different kinds, Parasaurophs from the one of the Jurassic Park movies have these dinosaurs, right? And we have this little guy, Dromaeosaurids, as well in this formation. Aside from all the dinosaurs, we also had diverse turtle ecology, as well as some crocodiles in that formation as well. And this one, so along with all the material, this slide has the stratigraphy of this quarry. So again, this quarry is toward the bottom of the formation in the like a bottom half of the middle member, right? And according to Roberts and others study in 2013, around this layer is dated as 76.42 million years ago, right? So that's the age. So we have nice, very refined age data for this rock formation. And in this section, we have some conglomerate overlain by the trough cross-laminated to cross-bedded sandstone, right? So that means this was deposited in stream, kind of like a crevasse spray type of deposits, right? So in that, we have some dinosaur fossils along with a bunch of fossils of turtles and crocodile teeth and so on, right? So the detail of this core is actually provided on the paper written by Titus, right? So I will just leave it as is for now. Right. So once you get the specimen, then my long journey kind of starts from there. So I use this dental drill to sample these isotope powdered specimen, right? And it's really tedious process. So I have to weigh about like 1 milligram or so specimen sample, right, for each data point. So I have to sit there hours and hours to make these powdered samples. Then from there, I have to use certain chemicals like bleach and hydrogen peroxide to remove diagenetic carbonate as well as the organic material, right? And I've lost a lot of specimens through this process too. So once that happens, you have to redo the previous step. So it is pretty time consuming. Right, and this is one of those days I was in the lab and drilling those specimens on my own, right? Right. But anyway, so that's one side of the story, and this process is for the carbonate samples, and I do different treatment for the phosphate sample, right? So instead of just cleaning we actually have to dissolve these minerals into an acid and reprecipitate this phosphate as a form of silver phosphate, right? So by doing this chemical conversion, I can get rid of all the other oxygen from this crystal, right? So the potentially contaminating carbonate as well as the hydroxide group on the OH at the very end of the chemical formula. So all those potential contaminations are removed by doing this. So that's part of the reason why some researchers claim this phosphate method is actually more reliable, right? And another reason to say that is, the bond between PO, the covalent bond in the phosphate ion, within the phosphate ion, is stronger than the carbonate and oxygen, carbon-oxygen bond in the carbonate ion, right? So for all those reasons combined, some researchers claim this phosphate phosphate method is better, right? But I wanted to see what story we can tell from the carbonate side as well. So that's the reason why I chose them both. Right. So once the samples are cleaned and processed, then these are analyzed. These samples are analyzed by using these advanced machines. That's the kind of machine I don't have in the community college now, but I still have some connection with the university, so I could ask them to analyze these for me, right? So in terms of the carbonate in the chamber, they add these specimen into acid. Like an acid, it's not a bath, but acid is added to these specimen to produce the CO2 gas, right? So the carbonate reacting with the acid, it's going to produce the CO2, and this is going to then analyzed by the mass spectrometer. So that's the procedure for the carbonate. But the phosphate is a little bit more demanding, right? So instead of just adding acid, this case we heating it up to the point that they break this bond between the phosphorus and the oxygen, right? So, but either way, this method right here is going to only tell you about the isotopic composition of the oxygen, whereas the carbonate would actually give you carbon and oxygen data, right? So that's the difference between those two techniques, right? And having done so, I have now I have the data, right? So I summarize all the data into some plots so you can actually visualize these differences, right? So the first diagram right here shows isotopic composition in terms of the carbon and oxygen in carbonate portion of the bone, right? Bone or the teeth, right? So the first thing you can probably pick up from this diagram is this the plus signs, right? So these are the data points for the dinosaur, tyrannosaurs, right? So these are distinctly different from the turtles and guards, right? So this is good news or good indicator for the actual biological data being preserved in these fossil materials, right? And also another thing you can see is the crocodile seem to separate themselves from the other aquatic animals, right? So even though they live in a similar environment, their isotopic data is different because of their different metabolism or the diet, right? So this is a good, kind of a good news, right? To proceed with this investigation. Right, so now I combine this oxygen data from the carbonate and phosphate and plot that on the 2D graph right here, right? So this black line right here, this is the theoretical equilibrium line, right? So, even though they precipitate this oxygen into different ions because of their metabolism, they would fall into somewhat like a close to 1-to-1 ratio with different intercept in isotopic composition, right? So having said that, the intercept might be actually different from different between the animals, right? So this, putting the same line for all the taxa is not necessarily a good idea, as you can kind of see on this dinosaur tooth example. But most of them fall into very close to this theoretical line. So that's another reason to say this data set is very reliable too for studying the paleoclimate, right? So again, we have another reason to support our data sets authenticity, really, right? Right, so having said that, now this is the main part of the study. So the variation within the tooth, right? So that would be potentially telling us about their seasonality. So this large tooth is actually most reliable data we have. So we have this really low point here and really high point here, right? So the sampling interval wasn't particularly great for this this tooth, but still what we can see is potential up and down, multiple ups and downs in their isotopic composition. So this might be telling us something about their seasonality in their environment, right? So all the small tooth, you know, they have much kind of coarser data points right here, right? And this because this, you know, the fragility of the specimen as well as, you know, this tiny drill, it didn't work quite well for these smaller teeth, but it worked great for this large tooth, right? So now we have potentially a seasonally driven isotopic difference in one tooth, right? So we can further analyze this data by using different conversion. In other words, mass, right? Right, so let's look into the water isotopic composition. Water isotopic composition estimated by these phosphate isotopic composition. Right, so again, we have carbonate and phosphate oxygen, right? And again, this carbonate might be less reliable in terms of their biological signal. And another reason to support that claim is actually this variation among the specimens I analyzed, right? So the standard deviation is somewhere between 0.11 to 0.88 for different groups of animals, right? Which is really small difference, right? And in comparison to that number, we have standard deviation from 1.0 to 1.6 for the phosphate specimens, right? So what that's telling me is, you know, the phosphate data again is more reliable compared to the carbonate ion, because again the phosphate ion is not as ubiquitous as carbonate ion. So some of these, you know, even though we have reason to believe, you know, at least some of it is biogenic signal, there's a reasonable doubt in this data set. So in order to study this water isotopic composition, I only focused on the phosphate data, right? Right, and then this is the converted number presented in the box plot, right? So statistically speaking, this again the tyrannosaur teeth are very different from all the other animals, right? So what that means is this dinosaur might be actually getting the water from a different source, right? Meaning they do not necessarily share the same ecological space, right? So that's a potential explanation, but another potential explanation is the migration, right? So we'll talk a little bit more about that in the following slides. Right, and I kind of forgot to, well, actually I didn't forget about it, but this conversion for this data is using all the equations previously shown on the earlier slides, right? Right. And now we can actually use this information from different animals. So the guards and turtle. Guards are those, you know, fish with really thick scale. And those ganoid scale again, is really highly mineralized. So we have a very reliable isotopic signature from these fish, right? And another thing to consider here is their metabolic difference. So turtles have some ways of regulating their body temperature, right? Bathing in the sun, getting out of the water, and so on. They can actually change their own body temperature, right? But the guards do not have that option. So what the isotope is really telling us here is that the turtle is more reliable indicator for the water isotope. And this by comparing this isotope data to the guards isotopic composition, we can actually use that for the temperature estimate, right? So the original study was actually published by this French scientist, Lecoyer, right? And by using the same equation, I estimated this mean temperature to be about 17.3 degrees Celsius, right? And also by seeing all the deviation in the data set, we can actually estimate the temperature range to be anywhere between 7.8 degrees to 25.1 degrees, right? So this number is a little bit lower than the previously proposed number, 20.2 degrees Celsius by Miller, right? But it still kind of falls in the similar range in terms of temperature. And also this temperature range kind of coincides with the study on the pedogenic carbonate by Bergner et al. In 2019, right? But then one thing I need to add here is the potential bias in the data set, right? So if you think about the turtles, they do not necessarily grow their shells during the winter time because they would be hibernating, right? So we are potentially biased in this temperature toward more summer temperature, and still end up with this very somewhat of a lower number than the Miller's temperature is suggesting. We need some additional study in future, right? And the last thing I want to add here is the precipitation isotopic composition, right? So again, by using the turtles and crocodiles, we can get a quite different range in their isotopic composition, right? So again, the tyrannosaur isotopic composition was much lower and had wider variation, right? And this potential cause for this variation is, again, the migration, right? So they might be migrating toward higher altitude in early spring or summertime, right? And that might actually create this lower isotopic signature due to closer proximity to the fresh snow melt, right? So that might drive their numbers down to this range, right? And also another thing we did not do is the serial sampling in the turtle shell. So if we actually consider this internal variation in the turtle shell, we might actually get a wider range in this sample too, right? Right. So all those information combined, we just compare these isotopic signature to known modern environments. So Sol and Fricke paper suggested this monsoonal environment was dominating this North America back in those days. So I chose Hanoi, Vietnam, and Brazil, the Cuiabá, Brazil, right? So in terms of their temperature and precipitation, there are some differences, but it kind of falls around the same range as the Kaiparowits estimate, right? So then these are the isotopic composition of those precipitation in the area, and the isotopic seasonal variation is somewhere between 6 to 10 per mil difference, right? So that fits fairly well with our proposed climate in Kaiparowits Formation, right? Right, and another thing to mention here is actually the metabolic difference, but we can kind of ignore that for now, right? Right, so using all these proxies, so instead of just focusing on one proxy, we can actually fill in some smaller detail in these paleoclimatic or the paleoenvironmental information, right? So this approach, the multi-proxy approach, is definitely one thing I would suggest to other researchers, right? And also this potential migration, this is something that requires further investigation. One paper written by Fricke did rule out the potential for moving to the south for the hadrosaurids, right? But this is something we need to investigate further, both in terms of isotope as well as actual fossil investigation, right? And also one way to increase our accuracy in the data is the finer sampling interval. So instead of just using the hand drill, we can use those machine-guided micro mill for the future study, right? Right, and for my own personal research, I would like to expand this study into the Hell Creek Formation, because now I live in Miles City, right? So the outcrop is only like an hour away. So this is my own future plan, right? So with that, I only have less than 10 minutes left here. So I'll leave the rest of the time for the question.
Great, thanks, Daigo. So folks out in the audience, if you have questions for Daigo, you can submit them in the Q&A box, which is found at the bottom of your screen. If you move your cursor over the bottom of the screen, that should pop up. You can send those in, and Daigo will do his best to tackle those. While we're waiting, I've got one question.
Sure.
So along the way, it occurred to me the idea of getting these teeth to address this question and try to figure out the paleoclimate from the past. Did you have the question in mind and then go digging for the teeth, or did you find the teeth on a dig and then realize you might be able to address this question? Or maybe it all kind of came.
Kind of like a mixture of both. So, as I got into the PhD program, the professor had potential research topics already. So, among those, I kind of started reading the paper and found this Kaiparowits Formation to be really fascinating. So one thing I didn't actually mention in the talk was the thickness of the formation is 860 meters thick. So that's like really rapid accumulation of the sediments. So from that information, I kind of realized, like a lot more information should be recorded in this rock formation compared to something like Hell Creek Formation, which is only 100 meters thick.
Okay.
So, but once I wander around in the field, then I come back with more questions every single time. So
yeah,
it's kind of a never-ending process if you really.
For sure, that's the truth.
Right.
Yeah. The more time you spend out there, the questions continue to evolve and change. That's neat.
Right.
And is that technique something that is well recognized, figured out, or was it something that you had to kind of figure out on your own in terms of getting enough of the sample to make the powder to do the analysis?
That's I did some revision to the previous technique, but it's very well established in European side of the research group. There are some other techniques involving like a laser abrasion kind of technique. So instead of just sampling everything and chemically process those, they can actually like ionize those crystals with the beam. And yeah, but that's like our university didn't have that kind of funding.
Sure.
So that's more advanced, and there's some debate about ionization messing with the isotopic composition. So it's still to be debated.
Okay.
Right. And I got one question here. So the question asked, do you want to find something else, like study different things on these fossils to find out more on what happened in the Cretaceous era? And the answer is definitely yes, because some of you might be familiar with the recent paper that just came out last month, I believe, about the Tanis, which is kind of a controversial site, but these researchers claim this fossil assemblage, it was actually created at the moment the asteroid hit the Earth. Right? So they have all the fossils jumbled up in part of the water column, as well as like branch piercing through the turtle and all kinds of like strange phenomenon. So yeah, that's something. You know, there's a potential for finding something like that in this part of the world. So yeah, I would love to continue field work and find something interesting, and hopefully apply my technique to those new assemblages. Because essentially this you know, new paper, you know, they use the same exact technique, stable isotope geochemistry, to narrow down the impact to be the springtime, right? At least in the northern hemisphere. So yeah, that's definitely something I would love to look into in the future.
Neat.
Right. So I got one question, and I don't see any more question at the moment.
Yeah, other questions from participants who've joined us? Anyone else? Daigo, I have just another question about your educational training. So is it a degree in paleontology? Is that?
So my undergraduate is actually paleontological option. So Montana State University has very in-depth paleontology coverage. So you can actually get the paleontology degree there, but my PhD is actually broader geoscience. So my department had anything from petroleum geology to something like isotope, like what I do. So
yeah, neat. Yeah, it's a interesting to take the chemistry side of it and apply it to paleontology for sure.
Right. Yeah, and that's something you know, I did not plan to do chemistry on the bone at the very beginning, but throughout the career, I kind of realized I'm better at chemistry than some other colleagues. So I'd as well just use that strength. And
yeah,
because, you know, you see a lot of smart people, so you have to find your own niche at some point.
Sure.
So that's how I ended up here. But yeah, there's another question here. So could you summarize your results quickly again, please? So my results or the interpretation, I don't know, but basically what the teeth show is the seasonal variation in terms of the isotopic signature. So yeah, those high number and low numbers corresponding to the isotopic signature of the precipitation, you know, high and low, right? So from that information, you can actually compare that to modern day data to, you know, interpret that as dry months and wet months, right? Basically summer and winter, right? So that's the kind of information you wouldn't get from other techniques like a leaf margin or the petrogenic carbonate, right? And in terms of the seasonal bias, you can get those from like different organisms because of the different metabolism. So it is always good to compare multiple organisms for actual broader climate context. Right. Hope that makes sense. Right.
Yeah, this is the interesting part of being on a Zoom webinar without the direct feedback.
Right. Yep.
Okay, so any last questions for Daigo? All right, well, we really appreciate you taking the time to put this together and share it with us. Interesting research. and fascinating to see what you can do. I've learned some about isotopic analyses and some of it that you can apply to understanding diets in different areas. But
Right.
this is a new one. This is interesting. Really, really neat.
Great. Thank you.
Yeah. So thanks very much. Hope we cross paths in the future, and take care. We appreciate it.
For sure. Great. Thank you so much.
Okay. Take care, everyone. Thanks very much for joining. Have a good evening.
You too.